Good afternoon. The subject of this talk will be how we model fibrotic lung disease using human pluripotent stem cells. Before I start, I'd like to thank the organizers for inviting me, and I would also like to divulge a potential conflict of interest. We have a sponsored research agreement with Lassen Therapeutics to study some of the drug targets that I will describe to you in this talk. So the focus of our research is a disease called idiopathic pulmonary fibrosis. It's a disease where the lung, in fact, undergoes a complete scarring, which leads to obliteration of alveoli, and ultimately patients die from hypoxia. This disease is not responsive to any immune suppression. It has a dismal prognosis, and the only curative treatment is lung transplantation. Two drugs, pirfenidone and nintedanib, have been developed that may slow disease progression but have not really been shown to extend the lives of patients. The incidence of the disease is increasing. There are about 20,000 to 30,000 deaths per year in the US. And a major impediment to further research in this disease is the fact that there are no really good animal models. Now, what do we know about the pathogenesis of idiopathic pulmonary fibrosis, or IPF as it is known? The predisposing factors are genetic, environmental, and age. Age is a major predisposing factor. Most patients are over 50 years old. There are likely environmental insults, such as smoking, inhalation of metal dust, and likely others. And there's a major genetic component. So mutations in surfactant genes in hPS-1, 2, and 4, and I will come back later in the talk to those three, in telomerase, and polymorphisms in a mucin called mucin 5B all seem to predispose to idiopathic pulmonary fibrosis. And when there's a single gene at the basis of this disease, we call it familial pulmonary fibrosis. It is believed that all of those conspire to cause epithelial injury, and the epithelial injury somehow causes proliferation of mesenchymal cells and deposition of collagen, which leads to the clinical pictures that we know, the clinical entity called IPF. Now, the questions in the field are, how do these insults cause epithelial injury? Which cells are really key in this injury process? And how do these injured cells cause proliferation of mesenchymal cells in the fibrotic process? And it is widely believed in the field that a specific cell type, the type 2 alveolar epithelial cell, plays a key role. Why do we think this? Because these cells express surfactant genes, and mutations in surfactant genes that cause proteotoxic stress are associated with very early occurring idiopathic pulmonary fibrosis. Now, the cell types in the lung and airways are many, and the key cell here in this disease is, as I mentioned, the type 2 alveolar epithelial cell, which sits in the alveoli, produces surfactants, which reduces the surface tension in the alveoli and keeps the alveoli open. This is the cell that's very likely very sensitive and susceptible to all kinds of injury. So what is the research strategy we follow? We have 10 years, for 10 years we have been studying pluripotent stem cells and how we can differentiate those pluripotent stem cells into lung and airway epithelial cells, and more specific into lung organoids containing epithelial cells and pulmonary mesenchyme. Now, what we do, and this is the strategy that I will walk you through during this talk, is we induce in these pluripotent stem cells mutations with a high penetrance for pulmonary fibrosis in humans. We make the lung organoids. We see that the lung organoids become fibrotic, as I will show you, and this allows us to study disease mechanisms and find drug targets in both familial and sporadic idiopathic pulmonary fibrosis. And in the last effort, we are trying to make large animal models for IPF, as small animal models, rodent models, do not faithfully recapitulate the disease. Pluripotent stem cells very likely don't require an introduction, but very briefly, we know of embryonic stem cells and induced pluripotent stem cells. Embryonic stem cells arise from the blastocyst of pre-implantation embryos. Induced pluripotent stem cells are obtained by reprogramming somatic cells to a state of pluripotency. Pluripotent stem cells can be differentiated into the three germ layers that arise after gastrulation of an embryo, and from these germ layers we can generate many mature tissues. This process is very challenging, however. So we have developed a system where we make lung organoids in matrigel from pluripotent stem cells. And without going into developmental and technical detail, what we basically do is we drive these pluripotent stem cells to become endoderm, the germ layer from which the lung is derived. Then we specify those to anterior foregut endoderm, which is the part of the endoderm that gives rise to the lung buds from which the lungs originate. This then gives rise to, in suspension culture, organoids that we call lung bud organoids. They are undifferentiated, equivalent to undifferentiated early anlage of the lung. And those we can plate in matrigel, and as you will see, this gives us branching organoids that contain lung cells and lung tissue. So this is an image of the lung bud organoids in suspension culture, and you will notice that they have these folding layers of cells, which are epithelial cells, as evidenced by the staining for EPCAM, FOXA2 and NKX2.1, and these two latter markers show that this is early pulmonary endoderm. The other cells express PDGF receptor alpha and are therefore mesenchymal. Now, when we separate those cells and perform RNA-seq, then you will see that on the EPCAM positive cells, we find expression of a variety of endodermal and early lung markers, in particular NKX2.1 and FOXA2, whereas in the EPCAM negative cells, the mesenchymal cells, we find a whole variety of markers of mesenchymal cells, and I point out here HOXB5 and TBX4, which are fairly specific within mesenchymal tissues for pulmonary mesenchyme. Now, when we plate these organoids in matrigel in the right growth factors, they will start budding and growing, and they develop these branching structures. And if you look at the arrow, you will see that something is happening there that looks very much like branching morphogenesis as it happens in a developing lung. At that time, these organoids begin to express a variety of more mature lung markers. I point out that all the cells express NKX2.1, a quintessential lung marker. The lumens of these organoids are decorated with mucin-1, which is a cell surface mucin expressed during lung development. We also find some evidence for mature cells, such as mucin-5AC for goblet cells, but most of the cells stain for surfactant protein C and surfactant protein B, which are markers for type 2 cells. You can also see here that these organoids are surrounded by very sparse mesenchymal tissues, as shown by staining for vimentin and CT90 or Thi1. These are Lightsheet microscopy images, which gives you a 3D idea of what these organoids look like. You will notice here in this middle image that most of the cells in the tips of the branching organoids express surfactant protein C, which qualifies them as type 2 alveoloepithelial cells, which make up the majority of these organoids. We benchmarked these organoids with respect to human development using publicly available databases that contain microarray data of most organs in the first trimester, second trimester of human gestation and in adults. And you will see here, green means a very good match, that our organoids show the best match with second trimester human lung. Now, to come back to IPF, I pointed out to you that mutation in certain genes are highly penetrant for the development of IPF, or rather of familial pulmonary fibrosis. And amongst these genes are the genes called hPS-1, 2, and 4. HPS stands for Hermansky-Pudlak Syndrome. This is a genetic disease of lysosome-related organelles. And the clinical features of these patients are listed here. These patients have pigmentation abnormalities because of defects in melanosomes, which are lysosome-related organelles. They have a bleeding disorder because of platelet dysfunction. The platelet dysfunction is caused by defects in delta granules. And finally, some genotypes develop pulmonary fibrosis because of abnormal lamellar body function. Lamellar bodies are also lysosome-related organelles and are responsible for the recycling and the secretion of surfactants. Now, interestingly, the mutations affect 4 complexes involved in lysosomal trafficking in mice and humans. There's block 1, block 2, block 3, and AP3. Only mutations that affect the block 3 and AP3 complexes cause pulmonary fibrosis, and in fact are amongst the most, when homozygous, the most penetrant mutations for this disease. So what we did is we introduced into yes cells mutations in HPS8 and non-fibrotic mutations, and mutations in hPS-1, 2, and 4, fibrotic mutations, and generated organoids from these. As you can see here, some of these mutations profoundly affected the morphology of the organoids. On the left are wild-type organoids and organoids with a mutation hPS-8, which is not clinically associated with fibrosis. On the right are organoids with mutations in hPS-1, hPS-2, and hPS-4, which are associated with fibrosis, and you will see that these organoids do not show the normal branching and seem to be enveloped in some other tissue. We also attempted to model heterozygous telomerase mutations. However, even in a heterozygous conformation, these embryonic stem cells were barely capable to generate any organoids. We further analyzed these organoids, and by staining, it's very striking that most markers of fibrosis are induced and upregulated in organoids with mutations that give rise to fibrosis in patients. And I point out in particular collagen 1 and 3, which are very, very strikingly present in those organoids. Collagen 1 and 3 and many of the other fibrotic markers are virtually absent in wild-type organoids and in hPS-8 mutant organoids. Shown here are the same data in Western blot, which basically shows that the organoid model can recapitulate the fibrotic risk in HPS patients. We next performed genome-wide RNA expression using the epithelial cells from these organoids. Why the epithelial cells? Because it's widely believed, as I explained previously, that the epithelial cells drive the fibrotic process. And we looked for genes that were, compared to wild type, differentially expressed in the fibrotic organoids and not in the control HPS organoid, hPS-8. And in this way, we could pull out a list of genes that was not very extensive, but very interesting. For starters, we found several genes that are known to be upregulated in the lungs of patients with fibrosis and or in rats or mice treated with bleomycin, which causes a fibrosis-like clinical entity. These are NEL1, PLA2G2A, and SERPIN1. SERPIN1, also known as PI1, has been known for more than 20 years as a marker of IPF. One gene that drew our attention, however, was interleukin-11, because it was very striking to find a hematopoietic cytokine expressed in the lung, to find it upregulated in fibrotic organoids, and furthermore, this is a gene that could be targeted because it is a soluble protein. Furthermore, consulting several publicly available RNAseq databases, both bulk and single-cell RNAseq of lungs of IPF patients, we found that IL-11 was almost always upregulated in every single study. Furthermore, we did in situ hybridization on lungs of IPF patients and patients with Hermansky-Pudlak Syndrome associated interstitial pneumonia, and found IL-11 to be widely expressed, including in type 2 cells and epithelial cells. This was not the case in lung samples that we got from surgical removal of neurothorax. Next, we wanted to prove that IL-11 is really critical to the fibrotic process. And to do this, we deleted the IL-11 gene using CRISPR-Cas9 gene editing from organoids containing a mutation hPS-4, which is also a mutation that gives rise to fibrosis in humans. And as you can see here, the hPS-4 organoids on the left-hand side are very abnormal and have extensive deposition of collagens 1 and 3. When these organoids do not express IL-11, or on the right-hand side of the IF panels, IL-11 receptor alpha, these fibrotic changes did not arise, and there was virtually no deposition of collagen 1 and 3. This shows that IL-11 is critical and required for fibrosis in the organoids. Furthermore, we added IL-11, recombinant IL-11, to wild-type organoids, and as you can see here, this induces fibrosis. The morphology of the organoids didn't change much, but there was clear deposition again of collagen 1 and 3, and by Western blot, accumulation of a variety of markers and proteins associated with mesenchymal proliferation and with fibrosis. So we conclude that interleukin-11 plays a critical role in fibrosis in the lung. And we've shown this by looking at the differential phenotypes of mutations, HPS-related mutations that are clinically associated with fibrosis versus mutations such as hPS-8, which are not associated with fibrosis. We furthermore also showed that deleting IDEL11 prevents fibrosis, whereas adding recombinant human IDEL11 induced fibrosis. After we published our paper around the same time, several other papers came out showing a role of IDEL11 in a variety of other fibrotic diseases. One of them is, again, idiopathic pulmonary fibrosis. Another one is cardiovascular fibrosis. A third one is non-alcoholic steatohepatitis. So interleukin-11 might play a general role in fibrosis, not just in the lung, but also in other organs. Now, what are we doing next? In addition to obviously trying to find out the mechanism of action of IDEL11 and to understand why, after stress to lamellar bodies in type 2 cell, IDEL11 is induced, we also want to develop an animal model for pulmonary fibrosis. The problem in the field is that mouse models with mutations in HPS genes do not show fibrosis. And so this is why we resorted to a larger animal model, namely the pig, to try to recapitulate the changes that we see in humans. And we chose the pig because the pigs have a cardiorespiratory physiology that's much more similar to that of humans than mice. We have generated, through targeting of zygotes with appropriate guide RNAs, for pigs with mutations in HPS1. We are currently analyzing the lungs of these pigs. These pigs have pigmentation abnormalities. They all have abnormal eyes, which is hard to see on this picture. And interestingly, one of the pigs is in fact hyperpigmented, and we don't know why that is, because HPS mutant humans have hypopigmentation. We are currently examining these pigs. And the workflow we are establishing is that we look for genes that are differentially regulated in fibrotic versus non-fibrotic organoids. We delete those in the organoids, see if the organoids become fibrotic or not, and then we will delete these genes on the HPS background in the pigs to see to what extent they affect the fibrotic process in vivo. Furthermore, we will test drug targets, and we're currently testing IDEL11 antibodies in the organoids, and if this works, we will very likely test those in the pigs as well. And the same strategy will follow for further disease drug targets we have developed. Finally, I would like to acknowledge the people who are involved in this work. The organoid model was established by Yawen Chen, who is now faculty at USC. And the fibrosis models were developed by Anna Chislak and Alex Trikoudis. Alex is currently at Regeneron, and in the lab, Tanya Timraj has taken over the torch here to continue this work. I'm very indebted to the pulmonary division at Columbia, in particular David Lederer, who is a world authority on IPF and has unfortunately also left Columbia, and Anjali Sadiq, who helped us with the lung samples and the human lung samples and the evaluation of the pathology of human lung samples. We're currently collaborating very intensely with the University of Missouri and the NSRRC there, who generated the HPS1 deficient pigs. This work was led by Kristin Whitworth and Randy Prater, and the pigs are now being shipped to the University of Iowa, where the lung team there, consisting of David Sauls, Mahmoud Abu Alaiwa, Michael Welsh, and Lokoy Brecht, will perform very detailed studies of the lungs of these pigs. Thank you very much for your attention.